The overall goal of this screen is to identify small molecule protein kinase inhibitors that modulate embryonic stem cell pluripotency.
This method can help answer key questions in the embryonic stem cell signaling field by identifying novel protein kinase signaling pathways that regulate embryonic stem cell pluripotency. So the main advantage of this technique is that the screen is relatively easy to perform using standard laboratory equipment and readily available reagents.
We first had the idea for this method when we realized that most research into pluripotency signaling pathways had completely ignored vast swathes of the kinome.
Prepare gelatin-coated 10 centimeter dishes by adding 5 milliliters of 0.1% gelatin to 10 centimeter plates. After incubating at room temperature for 5 minutes, aspirate the gelatin and let the plate dry for 2 minutes. Then, plate any standard mouse ES cell line on the coated dishes in standard mouse ES cell media containing 100 nanograms per milliliter of GST-tagged LIF, 10% fetal calf serum, and 5% knockout serum replacement. Culture the cells at 37 degrees Celsius, 5% CO2, and replace the media every day. Passage the mouse ES cells at around 80% confluency every second day by aspirating the media and washing with 5 milliliters of phosphate-buffered saline per plate. Then, add 1 milliliter of trypsin EDTA per plate of mouse ES cells and incubate at 37 degrees Celsius for 10 minutes. Following incubation, resuspend the trypsinized cells in 4 milliliters of media.